Great. So, hi everyone. My name is Dr. Emma Scott, and I'm going to talk to you today, as Laura said, about how we use sugar groups to diagnose and treat cancer, and in particular, prostate cancer. I thought that I would quickly start by giving you an introduction to myself and a bit about my background. I'm not a medic. I'm a research fellow here at Newcastle, and I'm funded by Prostate Cancer UK. What that means is that I receive funding from Prostate Cancer UK to carry out my own research, where I'm trying to develop better treatments for prostate cancer. I'm a member of a much wider research group, so this is my group here, and this is the Cancer Glycosylation group. And we work down at the Centre for Life, it used to be the Institute for Genetic Medicine, it's now the Biosciences Institute here in Newcastle. And before I was doing this, about 10 years ago, I was in a similar position to what many of you will be. So I was leaving sixth form college, and I went off to Liverpool University, where I studied biomedical genetics. Did that for 3 years and absolutely loved it. So I stayed and did a master's. And that was where I did a placement at the Northwest Cancer Research Centre, where I was looking at head and neck cancer. And that's when I got really interested in cancer research. So I came back to Newcastle in 2015 and I started a PhD where I was looking at how genes can influence liver disease and liver cancer and the influence that obesity can have on that. So that takes us up to today, where I joined my current research group and I started working on prostate cancer. So as I mentioned, I'm funded by Prostate Cancer UK, and the aim is ultimately to try and develop new treatments for prostate cancer. But why do we do it? Every single year, over 50,000 new cases of prostate cancer are diagnosed, and 11,000 men die every single year. It's the most common male malignancy in the UK, and approximately 1 in 8 men will be diagnosed with prostate cancer in their lifetime. It's often been thought of as a disease of kind of older men, and that's because if you're over 50, you do have an increased risk of developing prostate cancer. But really sadly, kind of prostate cancer has been overlooked for many years, and historically, for like kind of 50 years ago, loads of the major cancer research charities were not interested in funding prostate cancer research. They thought that it was just a disease of older men and they didn't really die from it. And that just absolutely is not the case. So Prostate Cancer UK over the past 20 years have done a phenomenal job of trying to raise funds to fund research that can really save lives. And they've done a brilliant job of that. And that's why I do what I do now. I thought I would give you an overview of how we diagnose prostate cancer at the moment. So what will happen is if a man is worried, he's displaying symptoms that he would like to be checked out, he'll go along to his GP. And more often than not, his GP will recommend a PSA test. And actually many men who are over the age of 50 will get PSA testing as standard. And what PSA testing is, it's a blood test where they're measuring PSA, which is prostate-specific antigen, and it's a protein which is secreted from the prostate into the blood. It's measured in a really quick and simple blood test, and high levels of PSA, as you can see here, can be a sign of prostate cancer. But there's some pretty major problems with PSA testing at the moment. So PSA levels can be high in men with cancer, but it isn't specific to this. And I'm sure with the current COVID-19 pandemic, a lot of you will have heard about the importance of sensitivity and specificity in diagnostic tests. So you'll all know that the PCR test for COVID is much more specific and sensitive than the lateral flows that we all do. And that makes it a much superior diagnostic test. And PSA is kind of like those lateral flows. PSA, it can be raised for all sorts of reasons. So PSA levels can be high if you've got an infection. It can be high, just if you've done a bit of exercise on the morning before you've gone and got your test, you can have elevated PSA levels. So for this reason, PSA can generate a lot of false positives. So men who get a positive test, but they don't actually have cancer. This is a major, major problem for prostate cancer, because as soon as you have an elevated PSA level, you'll be referred for more invasive, much more uncomfortable tests. They include a digital rectal exam and a prostate biopsy, as well as being uncomfortable. They can be quite painful, and also they carry their own risks of kind of infection, particularly with the prostate biopsy. I also want you to try and consider the kind of mental effect that that has on patients. So to get results back from a digital rectal exam and then a biopsy, that can take upward of 2 weeks and longer than that before they get the results back. And can you imagine living for 2 weeks or more worried that you have cancer every single day, to then be told that actually everything was fine? It really, really reduces the quality of life of patients. And that isn't the only problem with PSA testing. So PSA tests don't actually identify all of the men who have prostate cancer. So there'll be some men who go and get a PSA test and then the doctor will say, yeah, no, it's come back negative, you're fine. They'll go on and later down the line be diagnosed with a cancer that they could have had treated earlier. And this is absolutely devastating for patients. So I actually do a lot of engagement work with patients where they come into the lab and I go out to support groups and I'll tell them a bit about my work and what I do, but they also will tell me about their concerns and their worries, and we feed that back into our research program. And without fail, every single time that I do an engagement event like this, I'm asked, why can we not have a screening program in the way that we have the breast cancer screening program or the ovarian cancer smear tests? Men don't have that option for prostate cancer, and they want to know why. And what it boils down to really is PSA testing is just not good enough to implement a screening program. So I just wanted to talk through this infographic, which is from CR UK, where they're trying to explain why we can't implement a screening program based on PSA testing. So if we started out with 1,000 patients who they didn't have any kind of symptoms, and we didn't have a screening program, like this is kind of what happens now, what will happen is 1,000 men with no symptoms, 68 of those will go on to be diagnosed with prostate cancer. Unfortunately, 7 of those men will die from prostate cancer. 61 will be treated and will survive, and that's amazing. But if we did have a screening program, out of those 1,000 men, what you would get is actually 88 men being diagnosed with prostate cancer. 7 of them would die from prostate cancer, but 81 would have been treated and survived. Fantastic. You have an additional 20 patients in that screening program who have been diagnosed with prostate cancer, but they've actually just picked up, it's a kind of tumor that isn't growing. It doesn't cause any problems to the patient. We call it an indolent tumor. And they wouldn't have ever known that they had this tumor, and they would never have had any problems. They're going to go on to have unnecessary treatments and a reduced quality of life, worrying every single day that that tumor is going to start to grow. The most important thing to note on here is that actually, if we implement a screening program based on PSA testing, no additional lives would be saved. We would only pick up cancers that don't need any treatment. But there is an urgent need to develop a better diagnostic test so that we could in the future put in a screening program based on a new diagnostic test. The other unmet need in prostate cancer is the need for better treatments, and this is what I'm particularly interested in. So from those men who are diagnosed with prostate cancer, how do we treat it? As I've mentioned, some of them will have a type of tumor which it isn't really growing, it'll never cause them any harm, and they would never know that they had it. And this is called an indolent tumor. These men will live for decades without any problems. And actually the survival rate for prostate cancer is pretty good. So 78% of men who are diagnosed with prostate cancer will survive their cancer for at least 10 years. So what will happen with these men is they're placed on something called active surveillance, where they're checked regularly by their doctors to make sure that that tumor is still fine, still dormant, causing no problems. So for the other men who have a more aggressive tumor, what will happen to them is they'll have some sort of primary therapy. And this is an initial treatment, often surgery or radiotherapy. For surgery, they'll have kind of that tumor removed or their whole prostate removed. And, some men will be totally cured with surgery and they'll go on and be fine. Others, really unfortunately will be fine for a while and then their cancer will come back. What will happen in most cases is that they'll be offered a hormone-based treatment. So I'm sure loads of you will have heard about testosterone. It's a male steroidal hormone. And what this does is it really fuels prostate cancer growth, and that's what tumors use to grow. So these hormone treatments, what they do is they starve the tumor of testosterone so that they just can't grow. It doesn't kill them, but it stops them from growing. And, these treatments, they tend to work, and they work often for quite a number of years. But again, in some cases, those tumors will become resistant and they'll begin to grow again, even though they don't have that testosterone fuel in it. So for those men, we call them castrate resistant. And really, the only options that they have available are things like chemotherapy. You'll all have heard of chemotherapy. It's kind of a sledgehammer approach to cancer. It's not very refined, and you have a lot of unfortunate side effects. And what will happen with those men is they'll use chemotherapy to manage the disease, but it'll never cure them. And so for men with castrate-resistant prostate cancer, they actually are incurable, and currently there are no treatments available for these men. So there's a huge unmet clinical need for better diagnosis and better treatment in prostate cancer. So onto the sugars. So our work is interested in sugar groups, and I'm not talking about the type of sugar that you eat when I'm talking about sugars. I'm talking about sugar groups or carbohydrate structures, which are called glycans. And for a very long time, people have ignored these sugar groups. They're very difficult to study, and we didn't really have the technology to be able to understand them. They've been massively overlooked, so much so that when I started my undergraduate degree in genetics and biochemistry 10 years ago, they were never mentioned throughout the entire course of my degree. And I actually didn't really hear anything about them until I finished my PhD in 2018. So they've been ignored hugely because people thought that they were just a protective coating, like you see on Smarties, a hard shell on the outside of every single cell, and it's just there to protect them. Over the past 20 years, some brilliant chemists and biochemists have developed new technologies, which means that we can now actually figure out what they do. And when they've done that, we know now that it's so much more complicated than that. And actually, if you look at a cell like that, the sugars on the outside are highly complicated, and you can get them in thousands of different kind of structures. And really, they come in all different shapes and sizes. It's really, really complicated. And we now think of it more like a language. So if you think of DNA as kind of a language which holds all of the kind of critical information for life, these sugar groups are actually more like that than we thought. So one thing that they're trying to tell you about is this. So I don't know if any of you recognize what these letters are and what they might relate to. They're actually blood groups. And I would wonder how many of you would actually know what your blood group is. So these enzymes which make the sugars, they can dictate and tell you what your blood group is. So for those of you who are blood group A out there, you have a specific type of enzyme which adds one type of sugar to all of the cells in your body. For those of you who are blood group B, you would have a completely different enzyme which adds a completely different sugar to every cell in your body. And for those of you who are AB, well, you have one copy that you get from your mom and one copy that you get from your dad. So you have a mixture of these sugars on the outside of your cell. And if you're O, you don't have any of these sugars. And what I just wanted to use this to highlight really how complicated they are. They're not just a protective coating. They hold really, really important information, which has kind of been critical for human health. So for example, in this example, understanding these sugar groups has actually provided the basis for safe blood transfusions, which has, saved how many lives since its invention. And it means that we can safely give a blood transfusion without rejection. It seems a bit daft, really, that we thought that these structures were just a decorative and protective coating. So another thing that it can tell you is whether or not you've got cancer. So when you are diagnosed with cancer, you'll often need a biopsy. So a doctor will take a biopsy, and then it'll be sent off to a pathologist where they'll look at the tissue in great detail, and they'll stain it. And what they're looking for is really particular chemical changes. And this right here is an example of a prostate tumor from a real patient. And that brown color are these sugar structures that we're looking at. And the pathologist can look at all kinds of proteins and sugar structures to look at whether or not you have a cancer and how aggressive it is. And over the past 50 years, scientists have looked at these tissues from all different types of cancers and tumors. And time again, they've noticed these changes in these sugar structures. Only when you look at tumors, these particular changes in sugar structures. We've seen it in breast cancer, prostate cancer, pancreatic cancer. The same patterns are coming up time and time again. So we're now at a point where it's really well accepted that these sugars are changed in cancer, but we're trying to understand actually what they do, why are they changing, and can we use that information to improve diagnosis and treatment? So for the past 5 years, our group, led by Dr. Jennifer Monkley, has been looking at these chemical changes in prostate cancer. So as I mentioned, these brown colours in these pictures are the sugar structures in prostate tumours. And we're really fortunate that lots of men, when they're unfortunately diagnosed with prostate cancer, when they get their biopsy, they're asked if they consent to us using their tissue in research. And luckily, loads of men do. And we've had thousands of patient tissues in the lab, which have been fixed in formaldehyde, and we've looked at them. And we've identified a map or a pattern of these sugar changes. The problem is that changes in the tissue we can't really use to improve diagnosis of prostate cancer. So if you think about it, men without tumours would have to have a biopsy for us to be able to have a look at these sugar structures using this kind of method. And it's not really ethical. It's hard enough to get men to go and get a simple blood test at the GP to check if they've got cancer. They're not going to go for an optional biopsy. So what we asked is actually PSA is secreted into the blood. Are these sugars being secreted into the blood, and could we develop a blood test which would be able to diagnose prostate cancer? And what we've actually found, because we've spent another kind of 2 years, I think, and we measured and looked at patient blood from thousands of patients, and we found that actually these sugars are in the blood, they're being secreted from the prostate, and they actually can detect whether or not a man has prostate cancer. And when you compare that to PSA testing, it's both more sensitive and specific. So it's actually a better diagnostic test than what's currently being used. That work took about 4 years just to do the basic testing to prove that you could use this type of measurement to detect prostate tumours. It's not as straightforward as, okay, we can do that, let's do that in the NHS. So actually, once you get to this stage, you need to find a way of getting it to patients. One of the most efficient ways of doing that is actually by commercializing it through a company. So Dr. Jennifer Monkley, who I was working with on the test, she is the lead investigator on the work, and she's done a fantastic job of actually setting up a company outside of Newcastle University to develop this test. And she's currently making a product which could use this test within EU regulations as a diagnostic assay. It's something which is, it was funded originally by Prostate Cancer UK, and it's completely backed by them. They sit on the board in the company, and they're trying to help us get it into the NHS. So the test uses the exact same methodology that the PSA test uses. So the NHS wouldn't need to buy any additional equipment to be able to do it, and staff wouldn't need to be retrained to be able to carry it out. It's something which we really, really hope will be adopted by the NHS in the future, and it would make a huge difference to those men who are diagnosed with prostate cancer. So the next thing is, so we know they're important, we know they're in prostate tumours. Could we use them to develop a completely brand new treatment? And so that's what we're trying to do now. So you might remember earlier that I showed you this schematic, and I said for these men here where they've got castrate-resistant disease, there's no treatment options available. They have chemotherapy and manage it. There's no cure. Well, people all over the world right now are frantically trying to develop a brand new treatment option for these men so that when they get diagnosed with castrate-resistant disease, they have an option. And many of you might have heard about a new type of cancer treatment called immunotherapy. And this is actually what I work on, and I wanted to quickly explain to you how it works. So you'll all know, particularly at the minute, how important your immune system is, and that immune cells are the white blood cells which are always whizzing around your body. They're looking for things that aren't quite right, such as like an infection, a bacterial infection, but also one of the things that they're always on the lookout for is cancer. And what will happen normally is that an immune cell will come over to a normal cell, and it'll look at what's on the outside of it, and it'll inspect it. And the normal cell will say, whoa, look, we're good here. There's nothing to see. The immune cell will then go, all right, yeah, and move on to the next cell. If that cell is a cancer cell, however, the immune cell, it'll get up close to it, and it'll be able to read things on the surface, which will set off alarm bells. If you're lucky, the immune cell will then recognize it as a danger, and it'll launch an attack onto the cell, it'll kill it, and it'll remove it. It's an amazing process, and it's something which is going on all the time in your body. Cancer cells, though, they're sneaky, and they know what that if they're going to survive, they're going to have to hide from the immune system. And so they've come up with all different kinds of ways to hide. Because we now understand it a little bit better, it's actually allowed for the invention of immunotherapy. Immunotherapy has been completely groundbreaking, and actually in 2018, the Nobel Prize for Physiology was awarded for its discovery. Immunotherapy, it refers to an umbrella term for any kind of drug or treatment which will give the immune system a little boost. That means that you can have more immune cells which are available to look for these cancer cells, or it means that when they do find a cancer, they can launch kind of bigger, better attacks to eradicate that tumor. These kind of treatments are changing the outcomes of several cancers, and particularly cancers like melanoma. And they can be completely curative for cancer patients. It's amazing. And we're really only at the beginning of the process. The potential is huge. Currently, there's over 3,000 active clinical trials which are looking at immunotherapy for the treatment of cancer. And actually, because it's relying on your own just natural immune system, there's fewer side effects than conventional cancer drugs. But I mean, it isn't good for everything. Immunotherapy has been really effective, as I said, for like melanoma, but other cancers, it just isn't working for. And prostate cancer is one of those cancers where it's not working very well. Out of hundreds of immunotherapy trials which are ongoing, only one drug has actually made it through to be approved for use in prostate cancer patients. It's a tiny proportion of those patients who can have pembrolizumab, which is that drug. And it's the equivalent of about 1,300 men each year out of the 40,000 or 50,000 now actually who are diagnosed. It's not good. Another problem with this drug is it's only approved for men who have a large amount of DNA mutations in their tumor. Now, to get this information, we need to take that biopsy, which they will be getting as part of the diagnostic pathway, but we need to extract DNA from it and send it off for DNA sequencing. It's a really technical process, and it's fairly expensive, albeit the cost is coming down every single year. It requires someone with the technical abilities to be able to isolate that DNA and run that through a sequencing platform, but it also requires a type of scientist called a bioinformatician to make sense of all of the data which is generated. It makes it really, really difficult to hunt down the small percentage of men who would actually respond to this type of treatment. And the main reason that people think immunotherapy isn't working very well is because immune cells just aren't very good at recognizing prostate cancer cells. So I actually think that immunotherapy can be used for prostate cancer, and it would be a lifeline for those men who have no other treatment options. We just need to tweak it slightly. And I think the answer to unlocking immunotherapy for prostate cancer is in those sugar groups. So these sugar groups, as I mentioned earlier, are really important for blood groups, and that's because they're really important for how the immune system recognizes your cells as being part of you. And what we know is that these sugars are really important on normal, perfectly fine, healthy cells, and a particular sugar called sialic acid is found on the surface of every single cell in your body. So every single cell, normal cells, will have sialic acid dotted around on their surface. And what happens is that when that immune cell comes over, it'll recognize that sialic acid and go, oh yeah, you're fine, I'm moving on to the next one. Cancer cells, though, they've cottoned onto it, and what they're now thinking is, all right, we'll cover ourselves in this sialic acid. We're going to put loads of it on the surface to hide in plain sight from the immune system. So what that means is actually that immune cell will come over, it will see all of these sialic acid molecules on the surface, and it'll think, all right, everything's okay here. I don't need to do anything about this, and move on. Instead of killing it, it'll allow the tumor to grow and expand. So if any of you have ever heard the phrase wolf in sheep's clothing, it's the perfect expression to describe this process. So the cancer cell is using that sialic acid to describe it, to disguise itself as a sheep rather than a wolf. So the field at the moment is now working on whether we can develop brand new drugs which are going to remove the sugar coating from the cancer cells, which they're using to hide. So there are naturally occurring enzymes which, instead of adding that sialic acid, they remove it, and they're called sialidases. And so now we're asking, can we use these enzymes as drugs to remove this layer? And the hope is that by stripping that layer off of cancer cells, will actually allow more immune cells to recognize that cancer is bad. So you'll have more of them there so that they can dispose of the tumor to hopefully eradicate it completely. And there's currently really good data coming out of the US, which is demonstrating that this approach can 100% work. So as many of you will know, before moving into human drug trials, we have to carry out work on cells in the lab. So we grow cells on plastic and we test drugs on there. And if drugs are looking like they might be working, we take them into an animal trial. And so what we've seen from this group in the States is they've grown breast cancer cells in these dishes, and then they've injected them into a mouse. And the reason that we need to use a mouse as a model for this is that actually those cells that are grown in the dish, if you give them an immunotherapy drug, there's not an immune system in that dish to be able to act on those cells. So we have to put them into a mouse to be able to see the effect. So they've injected the cells into a mouse, and then they've left them to develop a solid breast cancer tumor. And what they've done once they saw tumors is they've given the mice a drug which targets that sialic acid and a drug which will boost that immune system and give it a little lift to be able to attack the tumors. And it's absolutely amazing, the data that they've generated. So they've seen that actually it's completely curative. So in 100% of the animals who were treated with this combination therapy, they saw a total reduction in their tumor burden. Imagine what that would mean for breast cancer patients. You know, if you could just give them these two drugs and everyone who was given it was cured. It's amazing. But they actually took the experiment a step further. And what they did was they took these mice that have no tumors because they've been cured, and then they injected them again with the exact same cancer cells which they injected the first time around. And they were totally expecting that these cells would form a tumor in the mouse as they always had done. Amazingly though, those no tumors were formed. They couldn't see a single tumor in the mouse. And what had happened is actually that the mouse's immune system had recognized that tumor cell as something that it had seen before, and it had destroyed it before it could even establish a tumor. The treatment, it allowed them to develop a memory of that cancer. And it means that if you imagine for patients, if they were cured of their cancer, but ever they had a recurrence or it came back, their immune system would be able to recognize that cancer, kill it, get rid of it before it even caused a problem for the patient. It's absolutely game changing. And it's something that we would really like to replicate for prostate cancer for those men who don't have any treatment options. So what we're currently working on in Newcastle at the moment is using patient cells, as I've said, grown in plastic, and patient tissue to answer the question specific for prostate cancer. Although that preclinical data is really promising for breast cancer patients, there's a lot of differences between different cancer types which dictate whether or not a treatment is suitable for a specific cancer and whether or not it'll work. Although both breast and prostate tumors have really high levels of that sialic acid, what I haven't really gone into is the fact that sialic acids can be slightly different. And actually, we've spent about 2 years now profiling in these cells and in that tissue the specific sialic acid which is expressed. So we've built up a really comprehensive map of all of the different sialic acids which are expressed in prostate tumors and where they are. The second thing which dictates whether or not a particular treatment is going to work and immunotherapy will work is the type of immune cells that are in that tumor. So not every tumor will have every type of immune cell. Immune cells can be very different. There's loads and loads of different subpopulations of them, and they all have different roles in the anti-cancer cycle. So what we've done, we've been profiling which immune cells we can find in those prostate tumors and what they do. And we found that actually the immune cells that are there are very different to the ones that are found in breast tumors. So we're really trying to tailor our approach towards something that will work for prostate cancer. So we've actually finished a huge amount of that really important kind of early stage work. And in the new year, we're going to be starting our own experiments where we're testing immunotherapy drugs in mouse models of prostate cancer. We're really hoping that we're going to see a reduction in the number in the prostate tumors in these mice, but it's really just the first step. we're going to be conducting lots of experiments to understand how these drugs are working, to generate enough data that hopefully we could take forward to show that a clinical trial in humans would be worthwhile. And then earlier I mentioned that actually there's only one immune checkpoint inhibitor which has been approved for use in prostate cancer. As it currently stands with FDA approval, only a finite amount of patients actually stand to benefit from this type of treatment. And it's really time intensive and expensive to be able to find out which men are actually going to benefit from this. As I've mentioned, we've carried out loads of work profiling in patient tissue and patient blood, all these different sugar groups. And, because we've done this in thousands of men, we actually know what percentage of men in prostate cancer have really high levels of sialic acid on their tumours. And by our calculations, it actually equates to 90% of those men who I mentioned had no treatment options available to them. That's a huge amount of men who could stand to benefit from this, as opposed to the very small amount of men who have lots of DNA mutations who currently would be prescribed pembrolizumab. What's even better though is actually because of the fantastic work that's being carried out by our company developing the diagnostic test, really soon we're going to have a commercially available test which is capable of easily identifying which men have really high levels of these sugars. And the test is much cheaper than sequencing. It's actually just about the same price as a current PSA test. And also it's relatively easy to perform. Like I say, we put a huge amount of effort into making sure that many of the biomedical scientists who currently work in the NHS would be able to carry out this test because they do it already routinely. They already have all of the equipment that they need and they have all the expertise. And so the ultimate aim really now is personalized medicine. So we would love to be able to get to a point where we can find a new immunotherapy based on these sugars that works in prostate cancer. And then we could combine that with our sugar-based diagnostic test to identify which men we can put forward for that specific treatment to give them the best possible outcome. So I'd like to leave it there and just say thank you to mainly Prostate Cancer UK for the amazing work that they've done for raising funds and funding this kind of research, and also the rest of my lab, particularly Jenny Monkley, who has been the lead investigator on a lot of the early work here. So I'm happy to answer any questions if you have any.
Thank you, Emma. That was so interesting, really, really interesting. We've had lots and lots of questions. So I have got a few to fire away, if that's okay.
Yeah, perfect. Go for it.
Perfect. So one of them is asking if it's true that positive pregnancy tests for men might indicate that there might have been some type of prostate cancer. I've never heard of this before.
I've never heard of this before. I don't think it would be, because I think it's a female hormone, isn't it, which is being used in pregnancy tests. Don't take this as like gospel, but I don't think it would be.
I'm starting with a hard question. Sorry.
And still go and get a PSA test if you get a positive pregnancy test.
Thank you. And there's a question here asking if prostate tumours wouldn't just show up on scans, but I'm guessing it's the cost of scans, but I'll let you take that.
Yeah, it is massively to do with the cost of scans, because MRI imaging is basically what we're using to try and improve it. But the problem with the imaging platforms is really expensive. And again, they want a screening program. Every single time we go and speak to patients, can we have a screening program? Can every man over the age of 45 go for a screen? And so MRI, it wouldn't be viable for the NHS. But also there's different problems with, depending on where you're imaging from. If you imagine the prostate's 3D, if there's a tumor on the back of it, you might miss it. But imaging, I think, is going to be kind of the way forward to improve the diagnostic pipeline. It still won't answer the question of a screening program, though. They really want that blood test that they could implement.
Excellent, thank you. And we had a few questions asking about basically the impact of the hormone therapy and what it does to kind of male characteristics.
It can be really, really awful. I think whenever we talk about prostate cancer, it's just, oh yeah, we'll give them anti-androgens and these hormone treatments. And they're thrown at prostate cancer patients, to be honest. And when I first started working on it, hadn't even considered the side effects that they go through. They go through hot flushes, they don't feel like men often. And it's, again, the same for some of the men who have a radical prostatectomy, so they have their whole prostate removed. They have the exact same problems. If you speak to them, though, if they've got an aggressive prostate cancer, they'd rather go down that route. But if we could, kind of take some of those men off of that and give them an immunotherapy that didn't have that kind of side effect, that would massively improve their quality of life.
I bet it would. Excellent, thank you. And are some blood groups more likely to develop cancers?
I don't think so, not that I know of. So the type of sugars that are dictating blood group, they're not sialic acid, they're a very different type of sugar. So not that I know of, I don't think so.
Thank you. And someone has asked, how did someone think to consider glycogens and investigate their changes related to cancer?
It's one of them things where people have investigated nearly everything, I think, in relation to cancer. It was just that we were profiling them. We profile everything in human tissue, and people were profiling it, and pathologists noticed time and time again they were seeing these sugars coming up in tumours, and it would have taken a while for them to put it together, but as soon as they did, we've profiled them in every type of tumour, and it's so striking that they change in every type of tumour. You kind of couldn't ignore it.
Thank you. And what is ADT and androgens?
So ADT, that's androgen deprivation therapy. So androgens is an umbrella term for the male steroid hormones. So testosterone is a very specific type of hormone, but it's an androgen. So androgens are an umbrella term for those type of ones. So ADT, androgen deprivation therapy, it's that therapy which starves the tumor of the testosterone, basically.
Excellent, thank you. And you may have touched on this, but there's a question asking if the sugar tests specific to prostate cancer, can that then be used to adapt for other cancers?
This is something which we're really interested in. It was actually very novel. For a long time, we were laughed at, myself and Jenny, when we said that we think that it would be detected in blood and that it might be secreted into the bloodstream. Everyone kind of laughed and said, why would it? That there would be no reason. So that was really novel and people hadn't looked at it. So it was only in 2019 that we actually patented the technology. And we've really tried to focus on prostate cancer, but I think we're now moving into pancreatic cancer, and there would be potential to do it for lots of, particularly like secretory cancers. So like liver cancer, secretory organs, I think would be really good for this type of test. But what you would have to do is, I said we spent 5 years profiling which sugars change before we even thought to look in the blood. That's going to be really specific to each type of cancer. So if you could profile in each one the sugars and then look into the blood, it would be, I would bet, a game changer for quite a few different cancers.
That sounds so exciting for the future, doesn't it? There's a lot of hope. That's fantastic. If the test for PSA levels can show as high or positive after somebody's done exercise, then how do people differentiate between prostate cancer and non-prostate cancer patients?
That's exactly the problem. Because there are loads of different reasons why it might be higher, and GPs, to be honest, they're not really very well equipped to deal with PSA testing. A lot of them will just give out a PSA test and then take it as gospel and say, oh, you don't have cancer, or you do have cancer. And that's the problem. These men are, they've gone for a jog in the morning, they've gone for their PSA test, it's come back high, they go into, as you can imagine, total panic, and they're then referred to a urologist who will do a digital rectal exam. And if things don't look right, or if they're not quite sure, they'll do a prostate biopsy, and it could be completely unnecessary. And that's why we can't use it as a screening test.
Excellent, thank you. And actually, we received about 5 or 6 versions of this question. So when the enzymes remove the silic acid from the cancer cells, why don't they also remove it from the non-cancer cells?
They do. Basically is the answer. It's not totally it's not very specific at the moment. What they're doing in breast cancer, I don't know if anyone's heard of HER2, but it's a receptor which is expressed by breast cells, and it's upregulated in breast cancer. And so what they've done is join that enzyme to a HER2 antibody so that it'll target it towards breast cancer cells preferentially. And we're playing around with the idea of doing that for prostate cancer, for looking at a protein which is really highly expressed by prostate cancer cells. Could we use that to make sure that it's actually targeted towards that kind of cancer cell rather than a normal cell?
And leading on from that, have you got any kind of concept of what the negative side effects could be of that drug?
Not yet. That's
No.
why we need to do the animal studies. There wasn't anything really in toxicology wise that came out of the breast cancer study, but that's really why we need to do, it's going to be about another 3 years of animal studies to try and understand what are the consequences of doing this. We have contingency plans so that if this doesn't work, the receptors on the immune cells which recognize that sialic acid, there's actually drugs which are approved for use to block those receptors. So as a backup plan, if this doesn't work, we're going to start blocking those receptors, and people already use those in humans anyway. So that might be something that we have to end up doing.
Fantastic. And why do you grow cells in plastic?
It's just where they like to grow. So that's what we do. So cancer cells, they grow just indefinitely. And if we want to do experiments on them, we have to have them in the lab. And you actually have to have them in really sterile conditions to stop bacteria, viruses getting into them. And so to put them in plastic, and they like stiff materials to be able to support themselves and to spread, to move, to proliferate. So plastic's the way forward for growing cancer cells.
Excellent. And are there other forms of testing available for cancer cells instead of testing on animals?
There's loads of different things. And normally what you would do is it's a kind of like a pipeline. So you would never go straight into an animal model. You would test things on cells. So we have tested our drugs on cells to see what happens, and we do see that it kills them sometimes. Like I said, the problem is we don't have an immune system there. So we've developed new models at Newcastle where you can isolate immune cells from people's blood. So people come in the lab and give a blood test, and we can isolate those immune cells and culture them with the cancer cells. But again, you don't have like the circulation, you don't have all the immune cells that you would have there. It's not the ideal model. And again, you can't kind of look for off-target effects, for side effects, for toxicology. And people are also growing kind of full organs in dishes, particularly prostate cancer, actually. We're growing prostate cancer organoids, but again, that grows the prostate tumor, but you don't have the immune system. So if things looked good in the cell lines, if things look good in that kind of full organ in a dish, that's when you would move forward to an animal model. You would never ever skip those steps before you move forward to that model.
Thank you. And at what age is a man likely to develop prostate cancer?
So there's not a specific age, but they recommend that someone over 50 gets tested. And it's kind of the older you get, the more your chance goes up of having prostate cancer. So anyone over the age of 50, speak to your dad, speak to your granddads. I would speak to them because men are awful at wanting to go and get a PSA test or to go and speak to the GP about it, because they're terrified that they're going to have a digital rectal exam or a prostate biopsy. Tell them to go and get a PSA test. It might not be the best test, but it's the best one that we've got at the moment. And I've spoken to patients who were in their early 40s and were diagnosed with aggressive prostate cancer, and it's devastating. So please speak to your dads and tell them to go to the GP.
Definitely. I think if anything's going to come out of this talk, hopefully that will be. Before this fully goes, I'm going to ask one more question. And what are the various routes of getting into research? So what was kind of what made you decide to want to do it, and how could other people do it?
So I really liked science at sixth form. I really liked biology. I liked chemistry. And I was always told, apply for medicine, do medicine. That's what you need to do. And I just loved science so much that I went and I spoke to someone who was a scientist and said, there's different ways to get into it. And do a degree that you enjoy in science. I did genetics, but you can do much more kind of broad courses. You can do a course in just biomedicine. You could do a course in biology. I work with people in the lab who just study biology at uni, and try and get as much research experience as you can. I hated my lectures, to be honest. They were nothing like what we do now. And as soon as I got into a lab, when I went into that Northwest Cancer Research Centre and started working in a lab, that was when I was like, wow, this is what I really, really want to do for the rest of my life. So try and speak to researchers, get into a lab, see what it's like. You don't have to have a master's, you don't have to have a PhD, you can just apply for a technician's job. As soon as you've got your undergraduate degree, it helps to do a PhD, but it's a really big undertaking if you do. But there's all kinds of ways. I would try and speak to someone, if you can, who works as a researcher.
Well, thank you very much, Emma. I'll end it now. Thank you for giving up your night, and hopefully I'll have this back by next week.
Thank you, Laura.
Thank you. Lovely. Thanks very much, Emma.
Bye, everyone. Thanks.